In this video, we're going to be looking at the diffusion of water across cell membranes, which is also called osmosis. Anytime we're talking about specifically the diffusion of water, that is referred to as osmosis. And then we'll look at how we can compare the strength of solutions as they compare to body fluids, the strength of the solution in body fluids. So here we go. So first, water can freely pass through a cell membrane, even though the interior, if we think about the bilayer, the lipid bilayer here, having a oily interior, a hydrophobic interior, water can pass right through the cell membrane thanks to the presence of these structures called aquaporins. These are proteins that create a channel that's always open that let water rush through the cell membrane, bypassing that hydrophobic interior, because the channel is completely lined with proteins that allow water to pass through. And this can happen very rapidly. According to one website I found, water can move through at the rate of more than 1 billion molecules of water passing through every second. So water can diffuse rapidly across cell membranes. And just like we were talking about the solutes being able to make their way across the cell membrane from high concentration to low concentration, moving from a region of very high concentration to a region of low concentration, water does the same thing. It also moves from a region of high concentration to a region of low concentration. So when we talk about cells or about solutions that we're giving to people in a healthcare setting, we always talk about the concentration of the solute. So when you make sense of osmosis, when you're trying to understand osmosis, you have to do a little bit of mental math, because the solute concentration is what we talk about, but it's the water concentration that controls the direction that water will diffuse as it's moving from a cell into the fluids around it, or from one cell to another cell. So let's say we have two cells. Cell A has 3% salt in it. Cell B has 10% salt solution in it. Which way is the water going to move? So the thing you have to figure out first is not what is the concentration of salt, but from the concentration of salt in the cells, what is the concentration of the water? So if we do a quick little bit of math, if a cell A here is 3% salt, well, we know that it must be then 97% water, while cell B, on the other hand, is 10% salt. So by definition of percentages, we know that it must be 90% water, because the two percentages are always going to have to add up to be 100%. So now the question is, which way will the water move? The salt can't move because it can't make its way through a semi-permeable membrane, but the water can because of all those aquaporins. So which direction does the water move? Think about it for a moment. Pause the video if you need to, and I'll see you in just a second. Okay, hopefully you have your answer. Let's see if it agrees with mine. So because there is a higher water concentration in cell A with 97% water compared to cell B with 90% water, we see water diffusing from cell A into cell B. So when we want to compare the strength of two solutions to each other, we do it by talking about the amount of solute that's found in each solution. If we have a cell, and so what we're seeing here is a cell in a water bath. Here's the cell, here's the water bath around it. If the concentration of salts in the cell and in the water bath is exactly the same, we talk about that as an isotonic solution. If, on the other hand, the water bath has more salt solute in it, then the water bath is a hypertonic solution when compared to the concentration of the cell. And we use the term hypertonic because hyper means above or greater than. The water bath solution is hypertonic to the cell solution because it has more of those salt molecules or salt ions. And then finally, if there is less salt in the water bath, then that is a solution that is hypotonic. Hypo means below rather than above. And so hypotonic solutions are those that have a lower concentration of salts than body fluids do. So isotonic, same concentration. Hypertonic, greater concentration of salts. Hypotonic, a lesser concentration of salts. Now we can also talk about other dissolved materials. It could be glucose that's dissolved. So in feeding bags, in IV bags that are used to feed, then they have a concentration of glucose in them, and so on. So any solute, you can talk about its tonicity by comparing it to the concentration of that solute inside of the cell. So let's think about the effects of different solutions on a living cell. Here we see an example where a red blood cell is placed into an isotonic solution. Because the membrane is selectively permeable and salts are unable to passively move from the cell into the solution, the only thing that can move is water, of course. But because the salt concentrations are equal or identical, the water concentrations in both the cell and in the solution that we put it into are also equal. And there is no net movement of water either into or out of the cell. So here we have a cell, and we're going to pretend now that the concentration inside the cell is 3% salt. So we've put it into an isotonic solution, and there is no net movement of water because both have a water concentration of 97%. Does that mean then that there is no water moving into the cell? No. There's always some water movement because the little collisions that happen with water colliding against water happen throughout the cell and throughout the solution. Some of them happen close enough to the cell that they're going to bump some water into the cell. But at the same time, other collisions are bumping an equal amount of water out of the cell. So there's no net movement of water. That means that the movement in and the movement out are equal, and so there is no change in the cell size. It neither shrinks nor swells. If, on the other hand, we put our cell into a hypertonic solution, once again, no movement of salts, but lots of movement of water. What's going to happen to our cell? So here is the cell. Again, we're going to pretend that the cell is 3% concentration of salts. That equals 97% concentration of water. A hypertonic solution has more dissolved salts in it. So let's pretend then that the hypertonic solution is 10%. So we have 97% water in the cell, only 90% water in the solution. And so we see a large amount of water moving out of the cell. This is like the egg that was placed into the Karo syrup when we were in lab. There's a greater concentration of water inside the cell, so water is forced out. Does that mean that there is no water coming in? Of course not. Water comes in too, but it's a smaller amount of water that comes in, so the net movement out of the cell causes plasmolysis of the cell, loss of the fluid from the plasma inside the cell, from the cytoplasm of the cell, and the cell becomes crenate. The cell goes from being nice and round and plump to being kind of shriveled or crenate. All right. Two conditions. The third condition would be putting it into the cell into a hypotonic solution. So here, if we think about the cell in a beaker again, like we were discussing in class, the cell is still 3%, but 97% water. But if the solution outside is hypotonic, maybe it only has 1% salt in it. That means that there's 99% water in the solution and only 97% in comparison inside the cell. Under these conditions, water will move from high concentration to low concentration, and we see a rapid influx of water into the cell. The cell becomes swollen. So our cell that normally has a little dimple in the middle of it, kind of giving it a solid, a donut shape that has a solid center, well, that cell becomes puffed up, becomes enlarged, puffed up with water because the water is going in. Now, as always, does this mean that there is no water leaving the cell? And again, the answer is of course not. But more water enters than leaves, leading to an overall net increase in water inside the cell. So it's the location with the highest concentration of salts that always has the lowest concentration of water. And so what we see is that water is always drawn toward the higher concentration of salt location in the body. So water is always drawn to the saltiest places because that's where the water pressure is lowest. And we talk about then the osmotic pressure of a solution. Osmotic meaning the pressure caused or the draw, the call of water into by osmosis into an area because of its high salt concentration. That's the osmotic pressure is highest where the solutions are saltiest. So it's not a push of water, but it's actually a pull or a call of water into a saltier region. That is the osmotic pressure. Okay, I think that's our last slide. Nope, almost done. So there is one other passive process that pushes or moves water from one location to another, or moves fluids from one location to another, and that is hydrostatic pressure. Hydrostatic pressure is found in all fluids, and it's caused by the effect of gravity, by the squeeze that the heart puts on the fluids, or by muscles squeezing on blood vessels and lymph vessels and creating pressure in those vessels that squeezes the blood or lymph to another location. So this type of fluid can force the fluid through the small gaps that are found between cells that sometimes that aren't completely attached to each other. So what we see, for instance, in blood vessels is that there are small gaps between the cells that when blood pressure pushes blood into a capillary that has these gaps, then fluid is pushed out into the tissues. So blood pressure causes fluid to be squeezed out. But if there are large elements like proteins dissolved in the blood or the red blood cells themselves, so we've got red blood cells, we've got these proteins that are dissolved in the blood, if they are too large to fit through the gaps, then filtration occurs. Just like filtration with your coffee, if you think about if you've got like a drip coffee maker, we use filtration. We fill up the coffee ground coffee filter with water. With coffee, and then hot water pours in, and then the pressure caused by gravity forces water to flow through the tiny pores in the paper. It's the same thing that happens in our capillaries. The pressure of the blood being pushed by the heart is what forces the blood, or the fluid in the blood, the water in the blood, out through the tiny crevices, the pores in the capillary walls. But all the blood cells and the large proteins found dissolved in our blood are kept inside. They're filtered so that only fluid leaves. And maybe really small nutrients like glucose might be able to slip out. Okay, so that's it for talking about osmosis and the tonicity, the comparisons of the strength of solutions, and then a little bit about filtration, the last of our passive processes. Any questions? Send me an email. Come to my office hours. Give me a phone call.